Hello everybody, my name is Emily Lorkheim, and today we're going to be discussing the decontamination of a COVID-19 vaccine production facility. Now I'm with Clordisis, and we helped do this decontamination in the United Kingdom in early 2021. So I'm going to give some background information as to why this decontamination had to take place. And some of it is that we needed to inactivate beta-lactams. So Alexander Fleming had discovered modern-day penicillin back in 1928, and the use of antibiotics spread vastly due to the benefits they provided, especially during wartime efforts. This spread very quickly. So the introduction of antibiotics was so dramatic that the life expectancy estimated at birth quickly changed from around 47 to upwards of 78 years old. And antibiotics work so well because of the powerful benefits of their beta-lactam ring. However, not all individuals have such a positive response to beta-lactam containing products. It's estimated that 3 to 10% of all individuals have an allergic response to penicillin alone. So these individuals often experience reactions like simple rashes to life-threatening anaphylaxis. Another possible reaction is blood pressure dropping to life-threatening levels, so this can cause things like lightheadedness and loss of consciousness. So because of this, the FDA requires a separation in manufacturing of non-beta-lactam containing drugs and beta-lactam containing drugs. The beta-lactam ring is the main component of several antibiotic families, the main ones being penicillin, cephalosporins, carbapenems, and monobactams. While the beta-lactam performs antibiotic mechanisms, the beta-lactam ring is also linked with the cause of anaphylaxis. This trait is the main reason for that the inactivation of beta-lactam antibiotics is necessary. So the risk. The U.S. Food and Drug Administration requires the detection of penicillin G and ampicillin residues in non-beta-lactam pharmaceuticals at a level of 0.03 ppm, so very low. This is severe restriction, so beta-lactam manufacturing facilities are often committed to production of beta-lactam products for the facility's life. This feature often results in equipment being discarded, even whole facilities being knocked down if they did want to manufacture something else in that same location. However, if we did have a means of inactivating the beta-lactam, we could offer a new opportunity for the facility to not only reuse equipment, but the building itself. So this can be a tremendous cost savings and a speed up in production. So there was a study conducted testing the efficacy of possibly using chlorine dioxide gas for the inactivation of beta-lactams. A major pharmaceutical firm had facilitated this because they had equipment that they wanted to repurpose. It had previously been used in beta-lactam manufacturing and they wanted to move away from it. So to explain a little bit about chlorine dioxide. Now, chlorine dioxide gas is a yellow-green gas, and it has an odor similar to chlorine, but nothing at all like it. This is a gentle sterilant, and it doesn't leave residues, and it doesn't harm materials. It's been recognized as a disinfectant since the early 1900s and has been approved in the U.S. EPA and Food and Drug Administration for many applications. And it's able to spread into an entire facility because it's a gas, just like oxygen. So the upper corners, the lower teeny scratches, everywhere the gas can spread to fill up an entire, millions of cubic feet of a building. Now, because it has such a small molecule size, it can get into those teeny areas. It's smaller than the smallest virus, which makes it excellent penetration. So chlorine dioxide acts as an oxidizing agent, and it reacts with several cellular constituents, including the cellular membrane of microbes. It has 5 electrons, so it has a great oxidation potential as well. And the oxidative attack on many proteins also prevents microorganisms from mutating into a resistant form. So what we did to study the efficacy of chlorine dioxide gas for the inactivation of beta-lactams was to take chemical indicators that were spiked with a cocktail of 8 beta-lactams. They were chosen to represent beta-lactams that were on the equipment as well as some of the more common ones that people see. So each chemical indicator had 5 ppm inoculated on the surface, and they were dried prior to the treatment with chlorine dioxide gas. So the pharmaceuticals manufacturer's requirement was a 3-log reduction, maximum post-exposure recovery of 0.1%, and that was the baseline for acceptance. So with approximately 5 ppm on each beta-lactam inoculated, the criteria 0.1% recovery would be equivalent to 0.005 ppm, or less than each beta-lactam, assuming that there was no loss on control CIs. So we encountered that a successful inactivation had occurred at a chlorine dioxide gas dosage of 7,240 ppm hours. So based on this, we can conclude that we had to reproduce a cycle of 30-minute conditioning phase at 75% relative humidity with an exposure of chlorine dioxide gas at least 7,240 ppm hours. Here you can see the various tests that were conducted, and the minimum of 7,240 ppm hours was deemed the baseline, and increasing dosages also resulted in death. So we know that that's the lowest we can use. So the location of the decontamination was in the United Kingdom, just west of London. So this facility, like I mentioned, they produce beta-lactam containing products, but because of the drastic nature of the COVID-19 pandemic and the need for a vaccine, they wanted to repurpose this facility to start producing the vaccine and spread it even quicker. So instead of rebuying a new property or starting new construction, they wanted to inactivate the beta-lactams instead. This facility had previously manufactured a product containing cephalosporins. So the inactivation process occurred with Chlordices, which is our company, as well as Cerecep, which is a company based out in the United Kingdom. And this occurred in March of 2021. The space that we were decontaminating in the facility was 247,203 cubic feet, and that entire space was filled with the chlorine dioxide gas. So what the chlorine dioxide gas process looks like is that there's a preconditioning phase where we raise humidity to levels between 60 and 75%. In this case, it was 75% to match that study. Conditioning, where we hold the relative humidity set point until it has been reached. Charge, which is the generation and delivery of chlorine dioxide gas. Exposure, which is the hold time once concentration has been reached, as well as aeration, which is the removal of chlorine dioxide gas from the chamber. So some supplies that were required in this particular instance are 2% chlorine gas cylinders. Chlordices manufactured CD cartridges, 1/4-inch red tubing, 1/4-inch green tubing, blowers, plastic sheeting, wall sealing paint, tape. Chlordices manufactured EMS with valve bank to monitor the process, carbon-based scrubbers, and an ATI portasense for chlorine dioxide gas monitoring. So this is the setup. We have the 2% chlorine cylinders that flow the 1/4-inch tubing right into the CD cartridges. The CD cartridges have a dry mixture of sodium chlorate and some other ingredients. Once that chlorine gas flows into those cylinders, we then produce the chlorine dioxide gas. And here's a setup. Those red tubes are this inject tubing. So that will flow from this staging area outside of the space into the space that we're gassing. The gas inject locations are evenly distributed throughout the space, and that's to get uniform distribution. And here's that inside the space. We also take samples of the gas. So all of the little green locations there are where we have two green tubes located, and we're pulling out samples every so often. It's continually pulling these, and that's to monitor what the concentration is at that point. We want to make sure that everywhere in the space has an equivalent concentration, or at minimum that it does reach the levels necessary that we're looking for. And then here you can see the sample tubing in the facility. So how we do this is by using our environmental monitoring system. So those sample locations pull those readings into the EMS. Through the photometer, we see what the concentration is and then calculate the dosage based off of it. So in this particular instance, we wanted to get a concentration of 5 milligrams per liter. And again, we want to reach that threshold of a dosage as well. Once all locations do reach the dosage, we know we did our job and we can aerate the space. So here we can see our concentration level. So all of those 12 sample points were trending very, very closely together. We reached that 5 milligram per level and we lowered it down to. So we're at around 5 o'clock, you can see the concentration lowering, and that's where aeration occurred. So all of the final dosages were over what we needed. We actually had an average of 11,795 ppm hours. And then you can see the dosages trending there as well. So aeration. We conducted the aeration through the use of a scrubber, and this occurred at around 4:54 p.m. And once there was a 0.0 ppm hour monitored on our ATI portasense when we walked through the facility thereafter, we were able to turn the space over. Now this ATI portasense will monitor also for leaks, but we want to make sure that after the space that even though we aerated properly, that we do in fact have a safe environment to turn over the facility to. So in conclusion, after the decontamination was completed, we set forth all goals accomplished, and we verified that there was no beta-lactams still active and present. So shortly thereafter, the pharmaceutical firm turned over the space and produced the COVID-19 vaccine there, and also held it as a storage spot for the vaccine. So I want to thank everybody again. Chlorine dioxide gas has been proven to inactivate beta-lactams, and we've used this time and time again. This particular instance was a very good cause, and we were very happy to help push that COVID-19 vaccine production site back into use so we can repurpose it. So I want to thank everybody very much, and if you have any questions or comments, feel free to call us or email us. Thank you again. Bye.